A very good morning. I am Lakshminarayana Gunta, NEET Zoology faculty at Super 60 Dupalvalsa, Srikakulam. In our previous session, we have discussed the phylum Porifera, Cnidaria and Ctenophora. And today in our session, we are going to discuss two more phyla, the Platyhelminthes and the Aschelminthes. The first one is Platyhelminthes. Platyhelminthes, the name itself indicates the flatness. They have dorso-ventrally flattened body, hence they are called as flatworms. Mostly they live as endoparasites in animals, including human beings. These are first bilaterally symmetrical triploblastic acoelomate animals. In our previous three phyla, we observed radial symmetry or asymmetry, whereas Platyhelminthes are the first animals which exhibit bilateral symmetry. Bilateral symmetry means the symmetry in which the animal can be cut into two equal halves or antimeres in only one plane that passes through the principal axis. Our humans also, we can be cut into two equal halves in this axis only. In any other axis, we can't be cut into two equal halves. These are the first triploblastic animals. All the three phylum that we have studied earlier, the Porifera, Cnidaria and Ctenophora, they are diploblastic animals. Means they have only two germ layers, the ectoderm and the endoderm. Here we have another layer, another embryonic layer, that is the mesoderm. The mesoderm, that's why these are the first triploblastic animals. And even though they are triploblastic animals, they are acoelomates, means they don't have any coelom, the body cavity. The body cavity is absent in these Platyhelminthes. Organ level and organ system level of organization is observed in these Platyhelminthes. Earlier we have seen only the tissue level or cellular level. Cellular level in Porifera, tissue level in Cnidaria and Ctenophora. And these tissues performing similar function, they form into an organ, and organs performing a similar function, they form into organ system. This organ level and organ system level of organization is first observed in Platyhelminthes. And the body is unsegmented. Segmentation you can see in earthworms, that is annelid, and arthropods, cockroaches, also in chordates and this body segmentation is absent in Platyhelminthes. Body is unsegmented. Some tapeworms like Taenia solium, Taenia saginata, they exhibit pseudometamerism. Here you can observe in this diagram, these are looking like metameres, but it is false metamerism. That's why it is called as pseudometamerism. And in parasitic Platyhelminthes or flatworms, hooks and suckers are observed. Here you can observe the hooks and suckers. These are hooks, suckers. These hooks and suckers are observed in parasitic forms, and digestive system, if present, is incomplete type of digestive system is observed in flatworms. That is called as incomplete gut. Means generally the alimentary canal, it starts with mouth and ends with anus. But here in Platyhelminthes or flatworms, only single opening is present, mouth is present, anus is absent. That's why it is called as incomplete gut. Incomplete digestive system is observed. And specialized excretory cells called as flame cells are observed in flatworms. Sexes are not separate in flatworms. That's why they are called as monoecious. Both male and female sexes, they are present in the same individual. And some members like planaria possess high degree of regeneration. Suppose if a planaria is cut into two parts, each part develops into a separate planaria. This type of high degree of regeneration is observed in Digestion. Fertilization is internal, that is, the fertilization takes place in the body, and development is indirect, involving a number of larvae like Meracidium, Sporocyst, Pedia, Cercaria, Metacercaria. Here you can observe a number of larval forms. These are all the larval forms of flatworms. This one is Meracidium. This one is radia, and this one is metacercaria, this one is cercaria, and this one is sporocyst. The examples includes Taenia solium, commonly called as the pork tapeworm, Taenia saginata, beef tapeworm, Echinococcus granulosus, dog tapeworm, Fasciola hepatica, sheep liver fluke, and Schistosoma hematobium, bilharzia, also called as bilharzia, which is a blood fluke. These are the examples for flatworms. Coming to the classification of Platyhelminthes. Phylum Platyhelminthes can be classified into three classes. Okay, the Turbellaria, the Trematoda, and the Cystoda. These are the three classes of phylum Platyhelminthes. Let us discuss one by one. The first one is Turbellaria. Class Turbellaria includes planarians, and mostly they are free living, and they can be seen in marine water, fresh water, even also terrestrial areas. Okay, the body is unsegmented. As they are free living one, there are no hooks and suckers. Example, the Digestion. Here you can observe the Digestion, and the second one is Convoluta. Digestion, Convoluta. These are the examples for Turbellaria. These hooks and suckers, they are absent in Turbellaria. As they are free living one, these hooks and suckers are absent in Turbellaria. Coming to the second class, that is Trematoda. Trematoda includes the flukes, sheep liver fluke, blood fluke. These are included in Trematoda, and these are parasitic on animals. The body is unsegmented, and oral and ventral sucker. This ventral sucker is also called as the acetabulum. And examples are Fasciola hepatica. This is Fasciola hepatica, sheep liver fluke, and Schistosoma hematobium, blood fluke. These are the examples for Trematoda. Oral and ventral suckers are observed, unlike that of Turbellaria, where suckers and hooks are absent, but in Trematoda, oral and ventral suckers are present. Ventral sucker is also called as the acetabulum. Okay, coming back to the third class, that is Cystoda. Cystoda includes tapeworms. All tapeworms are included in Cystoda. These are all endoparasites, and pseudometamerism is observed in this Cystoda. Here in the diagram you can observe the pseudometamerism. Even though they are looking like metameres, but this metamerism is false metamerism. It is not a true metamerism like that of annelids and arthropods. And here in this diagram you can observe the scolex and the proglottids. This one, the anteriormost region is called as the scolex. Here you can observe the hooks, and these are the suckers. The oral sucker and the ventral sucker. Ventral suckers are called as the acetabulum. And scolex bears hooks and suckers. Along with these hooks and suckers, it also bears bothriodia, leaf-like structures. Leaf-like structures are horseshoe-shaped structures are called as bothriodia. These bothriodia are observed in class Cystoda. These bothriodia and hooks and suckers, these are all useful for anchorage. As they are endoparasites, they need anchorage. They need to attach with the alimentary canal. And the examples include Taenia solium, called as the pork tapeworm. Taenia saginata is called as the beef tapeworm. Echinococcus granulosus is also called as the dog tapeworm. These are the examples of Cystoda. And coming back to next phylum, Aschelminthes. Aschelminthes, they are also called as Nematihelminthes, and they are also called as the roundworms because of their body which is circular in nature, and hence the name roundworms. They are also bilaterally symmetrical and triploblastic as like that of Platyhelminthes, but the difference is the Platyhelminthes, they are acoelomates, and these Aschelminthes, they are pseudocoelomates. And tube within tube body plan is observed for the first time in Aschelminthes. In order to protect themselves from the enzymatic action of the host, their body is covered by transparent, tough and protective cuticle which is made up of a collagenous protein, and they have complete alimentary canal. Unlike that of Platyhelminthes, those have incomplete gut or incomplete alimentary canal. These Aschelminthes, they have complete alimentary canal, and for the process of excretion they have specialized cells, H-shaped cells, H-shaped renate glands are there, H-shaped renate glands for excretion, and they have specialized structures called as amphids and phasmids. These amphids, they are cuticular depressions around the oral region performing chemoreceptor function, whereas the phasmids, they are present on the posterior portion and they are the glandulosensory structures. Okay, A for anterior. Remember that A means anterior side, P means posterior side. Amphids, they are present on the anterior region, phasmids, they are present on the posterior region. Amphids are chemoreceptor in function, phasmids are glandulosensory structures. Okay, next the sexes are separate. Unlike that of Platyhelminthes, here in Aschelminthes or Nematohelminthes, the sexes are separate. That's why these are called as dioecious. And sexual dimorphism is also seen. Means males and females, they are clearly demarcated. Generally the males, they are shorter than the females, and the females, they are longer and larger. The males, they have a curved posterior end, whereas females, they have a straight posterior end. Fertilization is internal within the body, and mostly they are oviparous. The examples are Ascaris lumbricoides. Some are ovoviviparous, like Wuchereria bancrofti. Ovoviviparity is a mode of reproduction in animals in which the embryos that develop inside the eggs, they remain in the mother's body until they are ready to hatch. This way, the Wuchereria bancrofti, they are ovoviviparous, and development may be direct or indirect. Coming to the classification of Aschelminthes, the Aschelminthes phylum is classified into two classes based on the presence or absence of the phasmids. Based on the presence or absence of the phasmids. If the phasmids, they are absent, they are included in the class Aphasmidia, and if the phasmids are present, they are included in Phasmidia. Aphasmidia. Aphasmids, A means no. Phasmids, they are not having any phasmids. The nematodes without phasmids are included in the class Aphasmidia. As the phasmids are absent, amphids are highly modified and the phasmids are absent. Examples for these Aphasmidia includes Trichinella, Trichina worm, this is Trichina worm, and Trichiurus lepturus, the whipworm. These are the examples for Aphasmidia. Coming back to the second class, Phasmidia. Phasmidia, as the name itself indicates the presence of phasmids. As the phasmids are present, the amphids are simple, and the examples includes Ascaris lumbricoides. This is an Ascaris male and this is an Ascaris female. The male has coiled posterior end, whereas the female has straight posterior end. Females are longer and larger than the males. Coming back to second example, the Ankylostoma duodenale, hookworm. It is called as hookworm. And the next example is Entrobius vermicularis, called as the pinworm. Entrobius vermicularis. And Wuchereria bancrofti. This Wuchereria bancrofti is also called as filarial worm. It is having a blunt anterior end and pointed posterior end. This Wuchereria is a major causative organism for this filariasis or elephantiasis. Okay, this finishes our Aschelminthes, and in our next session we are going to discuss the Annelida and Arthropoda. Thank you for watching.